It is in the Parc Naturel des Volcans d'Auvergne that is located the INRAE Research Centre of Clermont-Auvergne-Rhône-Alpes. In this centre, the Herbivores Research Unit studies the sustainability of ruminants production systems with a particular interest for grassland production systems. The worldwide challenge for tomorrow's livestock is to produce more and better with less, in order to feed the planet and satisfy the growing demand for animal products supposed to increase 50% by 2050. In this global context, ruminants play a major role by utilising forages, a resource non-competing, to produce milk and meat for human consumption, and maintaining landscape and its biodiversity. But besides, ruminants are targeted for their strong contribution to global warming through the greenhouse gas emissions. Methane is a greenhouse gas 28 times more powerful than CO2, but has also a much shorter lifetime in the atmosphere than CO2, 12 years versus 100 years, which makes methane an attractive amelioration target for short-term gains in global warming abatement. Ruminants are large producers of methane, which is mainly produced in the rumen and eliminated by eructation into the atmosphere. Methane accounts for 60% of the greenhouse gas emissions at farm level and 6% of the gross energy intake by the animal as energy loss. The objective of our research is to mitigate the methane emissions by ruminants while optimizing their digestive and production efficiency. Our research strategy is organized around 3 keywords: understanding, quantifying, and predicting. To understand, we study the mechanisms of methane production in the rumen to design strategies of methane mitigation. To predict, we look for peripheral indicators or predictors of methane emissions. The third keyword, quantifying, is the keystone of our research strategy, since quantification of methane emissions will allow us to test both the relevance of the strategies proposed to mitigate methane and the relevance of the predictors of methane emissions to screen a large number of animals in the field. Our research group has the expertise and tools to quantify methane emissions by using 3 different methods: the respiration chambers, the SF6 tracer technique, and the green feed system. The method of the respiration chambers is the most accurate and is considered as the gold standard method by the international scientific community. Why? Because it's a direct method which measures the totality of the methane produced by the animal. It is an automatic system allowing the continuous measurement of gas emissions. By this way, kinetics are measured in real time. In practice, the animal is within a chamber which is ventilated by an airflow controlled and measured. Methane concentrations inside the chamber are calculated from the difference between the concentration in the outgoing air and the incoming air. Then, the methane emission is calculated from the airflow rate and CH4 concentration in the gas collected. The tracer technique uses a gas named SF6 for sulfur hexafluoride. The SF6 is considered as an ideal tracer gas because it has the same behavior as methane and is not toxic for the rumen microbes or for the animal. This technique is based on the dilution of the SF6 within the other gases of fermentation in the rumen. This is an indirect method compared to the respiration chambers because we collect only a representative fraction of the gas expired. In practice, a small permeation tube is loaded with SF6 and calibrated by weighing during several weeks in the lab in order to determine the release flow rate of SF6 from the bolus. Then the bolus is inserted into the rumen and a breath sample is continuously collected around the animal's nostrils into a canister via a sampling device, composed of a capillary tube fitted to a halter and connected to a pre-evacuated canister. After collection of the breath gas, the concentrations of SF6 and methane in the canister are determined by gas chromatography. Daily methane production is calculated using the known flow rate of SF6 from the bolus and the concentration of SF6 and methane in the breath sample, corrected for ambient air. The green feed system was developed a few years ago by CLOC, an American company, and is now more and more used around the world. It's an automated system able to do spot measurements of methane emissions when the animal visits the feeder station. In practice, at each visit, the animal is identified by its electronic ear tag and receives a small amount of concentrate feed. When the animal eats, all the gas expired is extracted into the system. Simultaneous gas concentration, airflow rate, and other parameters are measured by different sensors of the system. Data is transmitted to a server where it is processed. The quantity of methane produced by the animal is calculated from the airflow rate and the methane concentration of the breath gas, corrected for ambient air. This calculation is done at each visit and cumulated for the day. Our research project requires a strong collaborative network including national and international scientists, technicians, students, and stakeholders of the ruminant sector. We disseminate our research by participating in national and international conferences, publishing articles, and patenting innovative data. We also contribute to science education and training of future youth through teaching and research.